I am delighted to introduce our final speaker, Uta Paszkowski, who leads the Serial Symbiosis Group at the Department of Plant Sciences at the University of Cambridge. She trained originally in plant pathology at the University of Cologne before receiving her PhD from ETH Zurich in biotechnology. And then she then spent time at the University of Basel and Syngenta in San Diego as a postdoc, where she deepened her expertise in plant biology and symbiosis. So since starting her independent career at the University of Lausanne, she has moved to the University of Cambridge, Cambridge University, where she now leads a world-renowned research team focused on plant microbe symbiosis and nutrient acquisition. We are delighted to add to the numerous distinctions Uta has received for her work the EMBO-sponsored keynote speaker for this meeting, and we're honored to have such a dynamic speaker help us cap off an excellent few days of plant biology. Uta.
Wow, what a wonderful set of 3 days. Thank you for putting it together, being amazing hosts, and thank you all for hanging in here until the very last speaker has completed and closed this meeting. Dynamic on the dance floor perhaps as well. Very good to see a meeting where dancing is still possible. Okay, now let's get on with it. So as is tradition for the EMBO keynote lectures, I first give a little bit of acknowledgement to EMBO. So I would like to mention that this keynote lecture is supported with funding from EMBO. And EMBO stands for the European Molecular Biology Organization. We have about 2,000 members. About 10% are from outside Europe, so the vast majority is from the European continent. And we admit approximately 60 new members every year. Now, I haven't done my homework. I need to see how I'm moving this forward. Yeah, good. And I'd like to draw your attention also to the multitude of different programs that EMBO is supporting. So from postdoctoral fellowships to grants that facilitate scientific exchange, courses, workshops, and then also young group leaders kind of meetings to facilitate getting your lab off the ground. And in addition, of course, we're all familiar with that EMBO also hosts a number of journals, including the EMBO Journal, EMBO Reports, Molecular Systems Biology, and so on and so forth. And they also support preprints and have created the Review Commons, which is a platform for peer review of preprints. So much for EMBO. Thank you, EMBO, for supporting this lecture. And I'd like to now move on to the science. So I've changed the title slightly and to give you a slightly more broader insight into one area of our research where we're studying the integration of arbuscular mycorrhizal symbiosis with plant phosphate nutrition. And we predominantly work in cereals, so rice is our main model plant species, but what we're looking at here, now I need to see the pointer, where is the pointer? Is it the big one? Oh, it's just a separate one. Okay, good. Thank you, Cyril. So what I'm showing here on this first slide is the key structure to the symbiosis. So you've had already a beautiful introduction earlier this morning from Lena and also from Evan. And here is one of the slides that I keep showing for decades in the tone of the meeting. So this is a picture from the 80s, but it very beautifully shows these tree-like structures that ironically the fungus forms inside the plant cell, inside plant cortex, root cortex cells. You see here the plant cell wall, so this absolutely sophisticated and fascinating structure is approximately 50 micron by 50 micron in height and width. So it's microscopically small. It is here where eventually the mutualism manifests, so the outcome of the symbiosis is indeed mutually beneficial, and we're putting our finger on where it happens here at these structures, where the fungus is nourished by the plant with organic carbon. The fungus is an obligate biotroph, it lacks fatty acid synthase, so it's entirely dependent on the plant for the supply of fatty acids to complete its life cycle. At the same time, the fungus is enhancing the mineral nutrition of the plant. It also provides water, but actually the effect is most pronounced for phosphate. And the benefit that plants have with regard to phosphate nutrition is burning down to this incredible increase of surface area contributed by the fungal mycelium for mineral, in particular phosphate acquisition. So what we in fact have is a conduit for minerals and water from these fungal hyphae through the fungal body all the way into the fungal structures inside the root and out of the arbuscules to the plant. So this is an incredibly effective route of nutrient uptake. Here we're looking at the summary of several years of radiotracer data that Eva Jacobsen in Denmark has compiled. And he was interested to actually quantify what is the percent contribution of the hypha pathway to overall phosphate nutrition of the plant, and he used over the years a set of really phylogenetically distant angiosperms, and in all cases we see this massive dominance of the symbiotic pathway over the direct pathway, with between 70 and eventually almost all 100% of the phosphate uptake being provided by that hyphal route. So, and given that we're looking at a really huge diversity of plant species here, if we extrapolate this to the fact that more than 80% of all land plant species engage in this interaction, and across all of the terrestrial ecosystems, we get an appreciation for what intense working horses these fungi are, because in the majority of these cases we would now be quite confident to assume that the phosphate acquisition of these the plants in these ecosystems is actually mediated via association with these mycorrhizal fungi, so that we quite fairly can conclude symbiotic plant nutrition is the default strategy in nature, and it is driving ecosystem productivity. So hugely important, and if we now want to stretch across from natural ecosystems to agroecosystems, we see that also here the symbiosis does make a significant difference. We're looking at beautiful work from Roy Soares that he conducted in Mexican median input subtropical fields. We have wild-type maize plants next to isogenic mutant plants that lack only one gene, rendering the mutant unable to engage in the symbiosis. You can make out with the naked eye that these plants are less happy than the wild type on the left-hand side. And remarkably, this is also translated ultimately in a difference of 30% in yield. So if you're able to engage with a vascular mycorrhizal fungi, you have a 30% yield increase. So it does have significant potential through data like this, which are increasing in the literature, for sustainable crop nutrition. So now, if we look across these multiple fascinating aspects of mycorrhizal symbiosis, from this incredible level of intimacy and compatibility when arbuscules form, all the way to a relevance in agroecosystems, we can say there's no way that we know enough about this symbiosis to understand the beauty of biology behind it, but also to perhaps make better use of it in agricultural settings. So we've learned a lot, and we saw some beautiful reflections of insights that have been gained this morning. But at the same time, there are challenges, or have been challenges, that we would love to master to get a deeper understanding of the processes that ultimately underpin development and also functioning of the symbiosis. And the big challenge to study, for example, if you're really interested in this avasculated cell stage, we cannot easily focus on studying that stage because the development of the symbiosis is A, highly dynamic, and B, absolutely non-synchronous, which is illustrated here. So on the left-hand side, you see a sort of summary of the different developmental stages, starting from a molecular crosstalk in the rhizosphere. Here, noteworthy is strigolactone as an exudate component plant hormone from roots that activates the fungus for symbiosis and also navigates the fungus towards the root. The fungus at the same time is releasing a whole cocktail of different effectors, proteins, and also small molecules, and also chitin oligosaccharides. And it is that molecular crosstalk that then reprograms both partners for that anticipated association. We then have surface contact formation of the fungus with the root. A hyphal swelling is produced, a hyphopodium, and then the fungus colonizes the root tissue, predominantly the outer root cell layers, the epidermis, the cortex. It does not breach the endodermis. It will never colonize the stele, and also not the meristematic tissue. And then as it arrives in the cortex, it will start to consecutively produce these arbuscles by invagination of the cortex plasma membrane, and through the iterative dichotomous branching. I think somebody is requested on the phone. Through this reiterative dichotomous branching, we build this beautiful tree-like structures that I showed you before that remain continuously surrounded by a plant-derived membrane, the so-called periarbuscular membrane, as you can appreciate here. And of course, this arbuscle development is accompanied by a monumental change in cell architecture. And foremost, really is to be mentioned this massive increase in membrane material that is produced to envelope the fungus. And despite this incredible investment from both partners to build these structures, they have a very short time lifespan. So they form and collapse within the course of just a few days. But it is here, as I said, where we're looking at the heart of mutualism. It is that bidirectional nutrient exchange that defines the mutualistic outcome. And as the fungus is well nourished, it then produces this fatty acid storage structures, vesicles, and also grows out into the soil to form this far-reaching mycelium, which is also where new fungal spores would form. And here on the right-hand side, you now can see that all of these stages that I just introduced you to really do occur at the same time, eventually in the same tissue. So to understand what happens at a specific stage, at a specific state of the cells that eventually engage with the fungus, we definitely have struggled in the past, but now see the opportunity through spatial transcriptomics, where we do get eventually the cellular resolution, or hope to get the cellular resolution of this complex interaction and across different stages on the whole tissue level. And we decided to work with the Resolve bioscience platform, so they use multiplex single molecule fluorescent in situ hybridization. So this is, if you wished, the early generation of spatial transcriptomic platforms. We had only 100 probes to design, so we predominantly focused on, call it a proof of concept, having markers for symbiosis, markers for cell types, and a few fungal genes as well. And Tanya decided to use longitudinal sections so as to capture eventually different stages of the interaction, and that worked really well in this beautiful section here. So you can see young growing arbuscles, mature arbuscles, and then also collapsing arbuscles. We have hyphae and we have vesicles. The stain here is DAPI for nuclei and wheat agglutinin for chitin. And you can see, you can also appreciate how the fungal nuclear distribution eventually looks like. We don't see fungal nuclei in small branches of the arbuscle. We see a lot in vesicles, and we can also appreciate that they are tiny in comparison with, for example, here a plant nucleus. So now let me introduce you to we have a ton of data, but I just picked very few, and some of you may have had a chance to talk to Gabriel at the poster already. So I would just make sure everybody takes home our highlights here. So we're looking here now at the first set of probes of genes. So basically, we have a constitutively expressed elongation factor gene from the fungus that very nicely allows to track fungal structures. You can see here the hyphae, the vesicles. If you were to just look at that channel, you would see the presence very nicely in the arbuscles. And then we focus here on the key transporters that are ultimately those transporters that define the bidirectional cost and benefit. We have the symbiotic phosphate transporter 11, we have the nitrate transporter, and we have one of the ABC half-size fatty acid efflux transporters. So mineral uptake and organic carbon efflux. And one of the key questions that also Resolve had was, can we actually detect transcripts of two different organisms in the same cell space? And the answer is clearly yes. You see in turquoise here the fungal elongation factor, and you see then evidence of all of the other plant transporters. So we can confirm through this method what we knew from the literature before, namely that these transporters are all arbuscle specifically expressed. But please also note this absolutely roaring amount of or abundance of PT11 transcript, and here we do a quantification, and please note that the y-axis is almost an order of magnitude different to the other transporters. And so phosphate, it comes back to phosphate over and over again as really the sort of massive currency of arbuscular mycorrhizal symbiosis. So now the fungus is pretty untractable genetically. We have no transformation system, and we know near to nothing, or flat and square, we know nothing about spatial expression patterns in the fungus. And here I'm just giving you one example case of some of those fungal genes. So here we have again our just DAPI and WGA stained section. So this is another section here now. And here we're looking at a kinase that has been published in the literature to be required for arbuscle development. And so we included it amongst the probe set. And what I would like to draw your attention to here is that there is an amazing, completely unexpected overlay of where we see that transcript, signal of that transcript, relative to the nuclei. So for that particular gene, there is that spatial correlation between fungal nuclei and transcript, which is not the case for other fungal genes. It's a bit hard to see here, but they adopt sort of distinct patterns, some being more expressed in the hyphae, all have some presence in arbuscles, and then this sort of amazing accumulation of this kinase transcript in the vesicles. So we definitely would want to know more about the fungus, and we can. And this brings me to my actually also second lab. So I have a second lab at Riken, Yokohama. And there we are focusing on the fungal side. And Akihiro Yamazaki is the deputy team leader in Japan. And we are now en route to, so this is a little bit of a future perspective of trying to get to the bottom of fungal transcriptional activity in a spatial context. And we would love to cover both asymbiotic and symbiotic stages. We are currently mining either our own or literature available bulk RNA-seq data. We have a lot for the symbiotic state, less so, but some for asymbiosis. And we've also recently succeeded, together with the team of Keiko Sugimoto, and here Akira Iwase is in the audience. So this is a fantastic team with working with Keiko on single nuclear RNA-seq from different plant tissue. And here's just an example case from tomato. We also have Marcantia, where we have been able for the first time to actually get single nuke fungal clusters. So this is going to be very insightful. We are hoping to do asymbiotic single nuke RNA-seq rather soon, so watch this space. This is going to be really, really exciting, and then we will select probes up to 500. We can select for a MERFISH-based spatial transcriptomic focusing on the fungus. So I'm really, really curious to see what we will find there. Okay, back to our symbiotic route. So I think one of the most exciting findings that we have made was, is shown here, and was that not all arbuscules seem to, or arbusculated cells, seem to express these typical markers for the symbiotic nutrient exchange, and you can see that sort of very easily. We have these two arbuscules here that show little to no signal. These are, again, this is our phosphate transporter, and then the rest of these genes, they all have something to do with fatty acid nourishment of the fungus. Or here again also the nitrate transporter. So you can see that there is not much happening in these two arbusculated cells. So first thought, of course, is this is an artifact. But then, as we looked through our different probes that we had, we found that indeed these two arbuscules show signal for other transcripts. Like here, it's the sweet and also the NOPE gene. And so it's not an artifact, and these arbuscules seem to indeed be just active in a different way. And looking across our different sections that we have, we found that there is a huge heterogeneity eventually to be found in the abundance of the different transcripts in the arbusculated cells. So that would be consistent with non-uniform functioning of these morphologically similar arbuscules. This is completely new. We've always stained typically with WGA, and you would not have anticipated that if you have arbuscules, especially like here, along a cell file, having such diversity eventually of the pattern. So this would be also consistent with the idea of having local tissue environments regulating eventually the transcriptional activity of the plant and also the fungus. So having said that, of course, one of the caveats of what we are doing here with spatial transcriptomics is that we get a snapshot, right? So this is a static state when you you introduce the static nature when you just prepare the tissue at whatever weeks post-inoculation. So what we need, what we're lacking from this, but what we need is the temporal solution. And ideally, we will be able to conduct non-invasive time-lapse imaging to really follow what happens, what are the spatial temporal dynamics of the proteins in the arbusculated cell as the arbuscle unfolds and also collapses. So Jen McGalley is the cell biologist in the lab, and she has developed this really nifty device, which she called AM Slide, so where you basically grow the plants in this small side compartment. The roots would extend into this foot here, and you can do live imaging as the plant grows. So we inoculate upon planting, and Jen has produced a whole set of different slides for different type of microscopes. And it works really beautifully. So here she did a every 24-hour shot of an area where arbuscles are developing, so that gives a little bit of an idea of what is eventually possible with that. You can also ignore mycorrhizal symbiosis and just use this device for any root cell biology real time. And Jen published this last year, and it's working by now quite well in a number of different labs. So Jen focused on initially on 3 different periavascular membrane intrinsic proteins. We have, once again, the symbiosis-specific phosphate transporter, these 2 half-size ABCG fatty acid efflux transporters, and then SCUMP is a protein that we use as a marker for the periavascular membrane. We know that it actually accompanies arbuscle development and collapse in the across all stages in the periavascular membrane. So in the first place, just looking at this sort of resolution that we can get if we do now non-invasive imaging. And we have a double reporter line here for the periavascular membrane, the marker SCUMP, and then we have our symbiotic phosphate transporter, and we go through different stages here of arbuscle life. So at the trunk stage, when the fungus is poking its hypha into the cortex cell, we see that there is a clear SCUMP signal, but actually PT11 cannot be detected. When we go to the young arbuscle, we see SCUMP at the trunk, at the coarse branches, but PT11 actually only coming up at the fine branches. Jen also did the intensity plots through the trunk, only the SCUMP membrane marker. Through the fine branches, then you see the presence of PT11 coming up, which is then very pronounced as we go into the fully unfolded arbuscles. And so this is known from the literature. The beautiful work from Maria Harrison here in upstate New York showed a long time ago that these kind of symbiosis-specific phosphate transporters do localize to the periavascular fine branch domain. So this was confirmatory, but then Jen also made this remarkable observation, which is coming back to our arbuscle individuality. So here we have a fully unfolded arbuscle, and here as well. Here we see PT11 wonderfully present, as I showed you on the previous slide. However, in that arbuscle, although it is equally well unfolded, we do not find much PT11 in the periavascular membrane. So once again, we see that there is a difference between individual arbuscles now with regard to protein transporter abundances on the periavascular membrane. And that would furthermore enhance that hypothesis that eventually the local context does actually play a role in regulating or fine-tuning the regulation of these nutrient transporters. So now, having this device at hand and being, of course, also interested in addressing the cost-benefit type of aspect of the nutrient exchange, it's been long-standing hypothesis in the community that the terms of trade are reciprocal. So you would expect that they are completely co-regulated. And here we're now looking at a triple reporter. So we have again our membrane marker, the phosphate transporter, and now the fatty acid carbon efflux transporters. And Jen came equipped with a mattress and a sleeping bag and took every 2 hours images for 54 hours. And here now, I mean, for us, this was a historic moment because it's the first time that we can actually really follow the arbuscles from birth to death. And you can see that here very nicely in that central cell where the arbuscle is flowering and then eventually very quickly also collapsing. So this now opens the door to a hell of a lot of different research questions. One of the first things that Jen determined was the lifespan. So in rice, Rhizophagus irregularis, has 2 to 4 days arbuscle lifespan, but actually super interesting I find is that arbuscles are never static. So it's not that they develop and sit there and pump in both directions. It is actually that these transporters are to be found on the periarbuscular membrane only during the phase of hyphal expansion. So we can then, in addition, of course, tease apart the spatial-temporal details, looking at individual frames. And one of the interesting observations that Jen made was that actually the carbon efflux to the fungus precedes the phosphate uptake transporter on the periarbuscular membrane, and so that would point to distinct spatiotemporal expression patterns and not really a strict co-regulation of these transporters. But as I said, the absence of a static state suggests that symbiotic phosphate uptake and fatty acid efflux is coupled to hyphal expansion, which is, I think, really neat. So this brings me to the summary of this part. So basically what I would like you to take home from this section is that, the symbiotic nutrient exchange is linked to dynamic arbuscle growth and it is locally regulated. So now I would like to switch to the second part, which is dealing more with, call it, systemic regulation of arbuscular mycorrhizal symbiosis, and I'm continuing here on the note that Evan started, namely that we know for a very long time, so this is from the late 60s, that there is an inverse correlation between the phosphate fertilization of the plants and the level of colonization. So, meaning that the plant quite precisely monitors its nutritional status, and when it's well nourished, it would not engage, or it would reduce the engagement with the fungus. So, we would love to know how this is eventually brought about, what are the mechanisms that regulate this high phosphate suppression, and I hope I'll be able to shed a little bit of light on that in the remaining 10-15 minutes. So it started off with a mutant that we identified in rice that had showed absolutely no interaction with the arbuscular mycorrhizal fungus. You see here the picture that you already know. The wild type shows all of these different fungal structures, and the mutant completely fails to engage with the fungus. Such a phenotype could be explained by two different processes. So either the mutant is unable to activate the fungus in an essential way, so the mutant would be unable to speak to the fungus, or alternatively unable to receive the fungus. So is it deaf or is it mute? And that could be answered when we looked at the transcriptome of wild type and mutant roots to fungal exudates, and we defined a diagnostic transcriptional signature in the wild type that was literally absent in the mutant, and that is consistent with the mutant having lost susceptibility to AM fungi. Now, the gene was eventually cloned. This was a many-year-long odyssey, and it's a long time ago, and it corresponds to dwarf 14-like, a pretty boring name, but that's what we had to stick to from the literature, and it actually corresponds to the karakin receptor. So the gene is very well characterized in Arabidopsis, and also in some other plants, but predominantly in Arabidopsis. So it is the receptor of karakin, which is a smoke constituent that fire-following plants require for the initiation of seed germination. So in Arabidopsis, it is famously called karakin insensitive 2. What is interesting to note is that it is the evolutionarily older paralog to the D14, the strigolactone receptor. So this is the D14-like, is the gene that we cloned. It was a bit of a surprise at the time. And Jong-Min Choi in the lab picked up from the knowledge that was generated in Arabidopsis, where we knew that the receptor D14-like, so Ki2 would interact with an F-box protein, D3 in rice, MUX2 in Arabidopsis, which would lead to the ubiquitination of a negative regulator, SMUX1, and sending it to the proteasome 26S proteasome, derepressing then these developmental programs, such as, for example, seed germination. So Zhongmin was able to confirm that the same receptor complex component are also required for symbiosis, as you see here. So we're looking at different fungal structures in the root, total, hyphopodia, intraradical hyphae, and arbuscles, and we can see that the D3 largely reproduces the D14-like phenotype. And when we then look at the mutation of the SMUX1 of the negative regulator, we see the colonization shooting up higher than wild type. So the removal of SMUX1 leads to that increased colonization consistent with SMUX1 operating as a negative regulator of symbiosis. And if you then bring SMUX1 into the background of D3, you find that the D3 mutant phenotype is suppressed, so SMUX1 operates downstream of D3 and D14-like, as previously shown in Arabidopsis in development. So we're dealing here with a classical hormonal double negative regulation, and these data suggested that the point of activating the D14-like receptor is to ultimately get rid of SMUX1. So that would imply that if we, when we see this low colonization in D14-like and D3, that should be attributable to a stabilization of SMUX1, and that is indeed the case. So you can see here that we have a very nice stabilization of SMUX1 in both of these mutants. But please also note that we actually do not detect SMUX1 at all in the wild type, and that would indicate that this pathway is just continuously on in the wild type, and that really changes our perspective at what to expect in terms of signaling to underpin conditioning plants for the symbiosis. We always think about you need activation, you need activation. You will need activation at one point, but what these data actually suggest is that perhaps having the pathway constitutively on and eventually having a possibility through stabilization of SMUX1 to block it might have been in evolutionary context what has happened, to evolve a pathway that allows to block colonization when conditions are less favorable. Okay, Zhongmin went ahead and then wanted to know transcriptionally what are the consequences of SMUX1 removal, and she found evidence for call it strigolactone signature. So basically the entire biosynthetic pathway of strigolactone was induced when SMUX1 was mutated, and we collaborated with Haro Baumeister to then measure abundance of strigolactone. And if I can draw your attention here to the amount of 4-deoxyorobanco in the D3 mutant compared to the double mutant with SMUX1, you can see and appreciate that there is a massive enhancement of, indeed, strigolactone production, and that showed back then for the first time that there's a crosstalk between the D14-like signaling and the strigolactone biosynthesis. The other signature that Zhongmin found was a symbiosis signaling signature. So just as a sort of rough guide, this is basically a summary of what we believe is happening when it comes to fungal recognition at the plasma membrane, secondary messenger being produced that leads ultimately to calcium spiking in the nucleus, and then this calcium signature is deciphered by a specific kinase, a calcium and calmodulin-dependent protein kinase, that then drives phosphorylation of relevant transcription factors to open the door for the fungus. So most of these components were transcriptionally induced in the SMUX1 mutant, which would point to the SMUX1 or the D14-like signaling pathway being upstream of the common symbiosis signaling pathway. And that is indeed the case. So we introduced a constitutively expressed autoactive version of this kinase in the D14-like mutant background and could indeed bring colonization back. So as a matter of fact, D14-like does operate, signaling does operate upstream of symbiosis signaling. So that leaves us with this model here, where we now find under favorable conditions that some unknown endogenous ligand is produced. So that is our favored hypothesis on the basis of this Western blot that I showed you before. D14-like being activated recruits the other components of the receptor complex, which leads to degradation of the negative regulator, liberating critical transcription factors for strigolactone biosynthesis and the common symbiosis signaling pathway, so basically calling the fungus with strigolactone and opening the doors for the fungus at the same time. And when we genetically remove SMAX1 from the system, we see this overshooting of colonization relative to the wild type. Under unfavorable conditions now, D14-like would not be activated because most likely the endogenous ligand is not produced, SMAX1 is stabilized, and we do not get colonization. So the big question now is, unfavorable conditions, does that actually eventually is the high phosphate suppression perhaps one of those unfavorable conditions? And that now brings Thomas into the game. He's a current postdoc in the lab who produced together with Zander Jones a ratiometric SMAX1 degron sensor. So you would have a constitutive strong promoter driving just the domain of the negative regulator that is known to be required for interaction with the receptor, and produce a translational fusion with red fluorescent protein. And then you have that ribosomal skipping sequence that eventually would lead to the RNA being read by the ribosome drops off, comes on again, and reads the rest of the RNA, so that you indeed get ratiometric levels of the one and the other fluorescent protein if both are stable at equal measures. So this allows us to really follow not expression of SMAX1 but stability of SMAX1, and the control for that is a deletion variant which has 4 amino acids deleted in that domain, which prevents the interaction with the receptor. Thomas introduced these constructs into protoplasts, and as expected we see the nuclear YFP signal, so this is NLS here tagged, so the nuclear YFP signal, little or nothing for the RFP. However, in the stabilized version, we now see a strong stabilization of SMAX1 in the nucleus. So the construct work, and many thanks to Zander Jones for helping us develop this. And then Thomas went ahead and did stable transformation. And now he's still in the process of characterizing these transgenic plants, but I'm introducing just some patterns that he has observed. So on the one hand, what you're looking at here is just a large lateral root of rice. And in terms of the signal, the hotter, the more stable the protein. And you can easily, with the naked eye, make out that there is a gradient. And when we do a cross-section, optical cross-section, we see that there is, in addition to the apical basal direction, also a proximal distal gradient of SMAX stability. SMAX is very stable in the endodermis. Wherever we looked. But then he also noticed some really interesting other patterns, like SMAX1 really is strongly stabilized in lateral root primordia. We're trying to zoom in. We don't know yet at the moment what comes first, but hopefully we'll be able to give an answer to that shortly. But we see from very early stages on that SMAX1 becomes stabilized. However, when the lateral root then grows out, SMAX1 is destabilized. So this is all very interesting. But now we of course want to know in how far SMAX1 becomes stabilized when we add high phosphate fertilization to the plant. So these are now data where Thomas quantified the nuclear signals of SMAX1 fluorescence in whole plants, and he looked at all three different root types, which in the end is not particularly relevant here. So we have crown roots, large laterals, fine laterals. The plants were grown at low phosphate or at high phosphate, and we have the wild type domain, or this, once again, the stabilized mutant domain. And what you can easily see when you just change the phosphate levels, that under high phosphate, SMAX indeed becomes more stabilized. You also can appreciate the stabilization that occurs when these 4 amino acids are deleted. But then what we also note is that there is a further stabilization here in the lateral roots, even in that deletion construct, which probably means that there is another pathway regulating SMAX1 stability in response to increased phosphate. So now we know SMAX1 is to some extent regulated by or stabilized by high phosphate. So if we turn that around now and say, okay, fine, if we grow plants under high phosphate where we see colonization is suppressed, in the mutant, SMAX1 mutant, do we see higher colonization? Because now we have removed SMAX1 from the system, and the answer is yes. So in the first place here we have wild type plants under low and high phosphate, and you can see the wild type is strongly suppressed in engaging with the fungus at high phosphate. However, in the case of the SMAX1 mutant, we see, while we still see some suppression, we also see that there is still a considerable colonization occurring. So we have now introduced SMAX1 CRISPR constructs into a variety of Japonica and Indica rice lines, and I'm just showing a few here. So we went into Eri 64 and then Bongsen. Bongsen is a very interesting indica variety. It's one of the most highly benefiting rice lines from colonization. So it grows, call it double as big when it is grown together with the fungus. We're here now having only high phosphate conditions, and we're comparing the wild type and the respective SMAX1, just one allele, but we have many. And so you can see that actually that relaxation of high phosphate suppression comes back in any of the rice cultivars that we so far have laid our hands on. So that means now that yes, indeed, unfavorable conditions to some extent really correspond to high phosphate conditions, and SMAX1, at least in part, but to a considerable part, explains this high phosphate suppression. And that's really, it's really a moment to appreciate the elegance of how nature has wired this whole system, because we have now one signaling pathway that integrates phosphate nutritional status of the plant with calling for help through strigolactone production and secretion, and at the same time opening the doors for mycorrhizal fungi to engage with. And that brings me to the end of the presentation. So I hope I was able to convince you that we have multiple levels of control, plant control of the engagement, and then probably with regard to the functioning, also the fine-tuning of the symbiosis, we have the D14-like signaling pathway that conditions plants for, in this case, rice, for symbiosis. And then we don't know yet what the controlling components are for this sort of tissue-specific fine-tuning of bidirectional nutrient transport, but this will be future areas of our research. This is the dream team. So I was talking to you about the work that Thomas and Gabriel as present members and Jen, present members in the lab, have done, and we've had fantastic former alumni in the lab. Nothing of this would be possible without our wonderful collaborations. Emma Wallington at the National Institute of Agricultural Botany does all of our rice transformation. Zander, huge thank you for the ratiometric sensor, Haro Baumeister strigolactone measurements, Keiko, Akira, Ayako for the single nuke collaboration. And yeah, thank you to our funders, and I'm happy to take questions.
Questions for Uta?
That was so cool. Thank you so much. I have two questions that might be related or might not be. The first one is, why do the arbusculars collapse? Is that, do we know, and is that maybe part of the sort of fungal benefit? And then do we know anything about the communication between cells or between arbusculars and neighboring cells? So do you think that the difference in timing that you're seeing is just the developmental time frame, or could they actually be signaling to one another?
Yeah, so with regard to the first question, why do they collapse? So this is why I was sort of emphasizing this idea of the nutrient transporters are only, the key nutrient transporters are only ever present on the periarbuscular membrane as long as the hyphae branch or elongate. At one point, there's no more space, right? So the fungus develops, and then collapses, and it usually collapses at about when it reaches the cell, the boundaries of the cell lumen. So I think there might be some form of regulation, of monitoring. The fungus is still, by the plant, still expanding, still delivering, still I can take up more of the desired nutrients, and once this is no longer happening, probably the plant pushes the fungus out. We have some trap data that would indicate where we could really dissect individual stages of the arbuscle development. Again, this was on Gabriel's poster. So taking a late arbuscle promoter, which is active from mature to the collapse stage, you see this massive induction of hydrolytic enzymes from the plant, a defense response. And it's interesting, there's a complete switch. This is off during the first bit. As the arbuscle develops, the defense response is off. As it kicks over, defense response comes on. So again, cause and consequence we don't know, but it might well be that the plant has a major control again at that level. The second question, so to say, within the fungus or within the plant? So, yeah, so the fungus is a syncytium. So it's which is one of the also another fascinating aspects. So if you look at these spatial transcriptomics data, and you see now, for example, one of these transcripts really localizes at around the nuclei. So that would tell us that there is some sort of microdomain within that fungal syncytium that is different from other areas where other transcripts are to be found. But communication within the fungus, or spatially really specific expression of any fungal transcript at any particular state, is a big conundrum, because the nuclei on the top are zipping through this through the cytoplasm. So in terms of cell-to-cell communication in the plant, so there's some genes that again Maria Harrison had identified that sort of almost labeled a cell file that is going to be colonized, so that precedes the fungal colonization. So for sure there is communication occurring. Which form this might take, who knows? Yeah. Thank you. Good questions.
That was fascinating. Thank you. It might be a little similar to Becky's question, but I was fascinated by the localization of the plant transporters on the mycorrhizal membrane. I was wondering if you looked into were there like motifs or patterns between the plant transporters that localized there?
So you mean zooming in further to periarbuscular membrane subdomains and see if they wholly overlap? Is that the question?
I think what I'm asking is what makes the plant transporters localized there?
Yeah. Trafficking. But yeah, so we know relatively little about how that is again controlled. So Maria has done most work in that realm, and she defined that the promoter activity, so there's really fine-tuned windows of promoter activity in the course of the arbuscle development is important to drive the expression of the transcript and then likely the translation right away into the right developmental window, because during that developmental window, sort of everything is channeled towards, that is a membrane intrinsic protein, towards this growing beast. No? So lots to discover there. We know very little about how that could be regulated. And she has, in support of that statement, so she has used transporters that normally would not localize to the periavascular membrane, other phosphate transporters, and drive them under this PT11 homologous promoter. And sure enough, also that transporter goes straight to the periavascular membrane.
Just amazing talk. Every single story was just wow. But I have a very simple question. Do you have any thought about the carrageen-like ligand for the coming from the avascular mycorrhiza?
I wish we would.
So you can profile something upon induction of the.
Yeah. So this was a project that Giles initiated in Cambridge to eventually hunt the KL, as it's called, the Ki2 ligand. And so far we don't have any news, and now Giles is thinking of other matters, so I'm not quite sure that we would continue this, because I think it will be difficult to get to the to that molecule in the first place. And what we've done alternatively in my lab, so I've collaborated with Shinya Hagihara and other colleagues at the chemistry campus of Riken and Waco, and we've done a chemical genomic screen.
I see.
And identified molecules that bind, initiate signaling, and enhance colonization, but we're not sure about specificity yet, so
that's exciting.
Hopefully, yeah.
Thank you.
We'll take one more question in the back there. Thanks.
Hi, thank you. That was a really amazing talk. I have maybe a basic question. So does all this happen below the Casparian strip on the root?
Can you speak up a little bit?
Does all this happen like below the Casparian strip on the root? Or is it possible for these arbuscular or these arbuscles to like recolonize a region that has been previously colonized and collapsed? Or,
I think it would be, we don't know, is the short answer. And rice has an exodermis as well, and a sclerenchymatic, sclerenchyma, so a heavily fortified cell layer in the outer layers of the root, so one after the other. So either the fungus is tickling the plant to loosen this up so that he can come through. What regards the endodermis, we don't need to breach the endodermis, so this can sit there quietly, this is fine, but the exodermis and the sclerenchyma in rice would be quite an obstacle if you had to get through. So it would be a wonderful project now that we have these different markers to create the respective lines, use the AM slide, and follow this all in real time. We can. So we should do things like that. We don't know if the fungus has a preferred zone, because whenever we look, we see that it's already sort of everywhere. No? So even if it starts to colonize, you would see it in different parts of the root. So I'm not quite sure what we are expecting. Most likely, we have to get through these outer cell layers before they are suberinized, lignified, whatever the current understanding is. Should be talking to Siobhan. Yeah.
All right, let's thank Uta one more time for a beautiful talk.
Thank you.